Very good morning to all the speakers and participants. Our honorable guest, Dr. Saiful Izwan Abdul Razak, the group leader of BioInspira Research Group, Frontier Material Research Alliance from University Technology Malaysia. Our speakers for today from Thailand, Dr. Suryan Supavanich and Dr. Chanpon Chawsap. Both are the assistant professors in the Department of Agricultural Education, Faculty of Industrial Education and Technology, King Mongkut Institute of Technology, Lat Krabang, Thailand. And our speakers from Malaysia, Dr. Razauddin Muhammad Zulkifli and TS Dr. Shazwani Itri Amran. Both are senior lecturers in the Department of Bioscience, Faculty of Science, University of Technology, Malaysia. Lecturers and fellows audience from Malaysia and Thailand. Before we begin our webinar for today, we would like to invite Dr. Saiful Izwan Abdul Razak, the group leader of BioInspira Research Group, Frontier Material Research Alliance, UTM, to officiate this webinar on future agricultural technology for 21st century. Please welcome Dr. Izwan.
Okay, Assalamualaikum and good morning. First of all, I would like to welcome all the participants from University Technology Malaysia and also King Mongkut University of Technology. Okay, and then I would like to thank Dr. Razauddin for organizing this nice event, Future Technology in Agriculture. Okay, we are from BioInspira Group, Bio-Inspired Device and Tissue Engineering. Okay, and our group is diverse in our research. Okay, we have researchers doing on agriculture, on tissue engineering, on biomechanics, on biomaterials, towards all applications in terms of bio device and also tissue engineering. Okay, and then in terms of agriculture, we also have some researchers doing on nutraceuticals, some food products, and also Dr. Izzah also doing some on honey research. In terms of agriculture, so we are doing some bioprinting of food and also bioprinting of meat. Maybe this can contribute a bit towards the research on agriculture. Okay, I'm once again I would like to thank Dr. Razauddin for organizing this nice event. I think that's all from me. Okay, thank you.
Okay, thank you very much, Dr. Saiful Izwan, for your great officiate and opening speech for our today's webinar. Ladies and gentlemen, we would like to welcome all of you to this webinar on future agricultural technology for 21st century. So now we will start our first session with our first respective speaker, Dr. Suryan Supavanich. Dr. Suryan Supavanich is an assistant professor who is specialized in post-harvest technology. He received his PhD in biosciences, nutritional science from the University of Nottingham, United Kingdom. Dr. Supavanich also had a postdoctoral fellowship experience at the University of Natural Resources and Life Sciences, Vienna, Australia in 2012 and 2015. Without further ado, please welcome Dr. Suryan Supavanich to deliver his talk about elicitors treatments, maintaining and improving post-harvest quality of fruits and vegetables. Please welcome Dr. Supavanich.
Okay, thank you everyone, and thank you for UTM that invited me to present my research that I have done so far in the criteria of the post-harvest technology. Today, I will present my work is related to the using the elicitor to maintain and improving qualities of the post-harvest horticultural crops. So anyone can see my presentation? Huh? Wait a minute. Anyone can see my presentation? Okay. Okay. What is an elicitor? Actually, elicitor is it can be anything that it can induce the secondary metabolites in the plants. Wait a minute. Okay. And the thing is including the chemical things, physical things, and also microbiological things. And we use the benefits that they induce the secondary metabolite in plant uh, to improve the plant's qualities and also improve the nutritional quality of the plants. And they also stimulate the uh, resistance to the certain physiological disorder of the fruit and vegetables after harvest and during storage. So what is the elicitor act to the plant? Actually, when the plant receives the elicitors, the elicitor, they will attack to the membrane, in the plasmatic membrane, and they got the receptor. And when the elicitor do the receptor, and then they send the signal and make the plant stimulate different response and also the genes expression that related to the secondary metabolite biosynthesis, and they can induce the bioactive compound, antioxidants, and also strengthening the cell membrane. So what kind of the elicitor that using in the post-harvest, we can separate into three groups. Is the one is a chemical elicitor. That's in my research, I'm interested in the chemical elicitor. The chemical elicitors such as the plant regulators, some vitamin like cyanocobalamin, vitamin B12, and any chemical that the plants receive and induce the secondary metabolite. And the second group is the physical elicitor. Physical elicitor that is we use in the post-harvest, such as the radiations, UV radiations, heat treatment, and also ultrasounds. For the biological elicitors, these some microbials such as the Penicillium, Candida, Collectoticum can induce the secondary metabolite in the plants as well. So we also use as the biological elicitor. But actually, in my research, I'm focused on the chemicals elicitor and physical elicitors. So the chemical elicitor that I have done so far is like plant molecular biologies. Plant regulators such as salicylic acid, methylgesmonase, and I'm so interested to use the new novel chemical is vitamin B12 as well. And then I will show how these chemical elicitor affect to the post-harvest fruit and vegetables. So for the both salicylic acid and methylgesmonate, we know they are the plant growth regulators that they release when the plant is subjected to the biotic and abiotic stresses. And the salicylic acids, actually for the food application, salicylic acid is inhibited because it's quite toxic to the people. But for the post-harvest use, we use very small concentration, and the small concentration is not affect to the human and environment at all. And from my experience, I have to check the salicylic acid content after treatment, and I didn't find any concentration of the salicylic acid after treatment. But normally the treatment that we use the concentration is lower than 2 millimole actually. So for the methylgesmonase, we know it's a volatile organic compound. It can produce when they got the stress, and they also induce the different mechanisms, and also in the many diverse developmental plant environmental pathways such as the seed germination, loose growth, flowering, fruit ripening, and also senescence. So this is the effect of salicylic acid. When the plant got the salicylic acid, the salicylic acid can induce antioxidant system in plants. So they also induce the different enzymes, and also they also decrease the lipid peroxidation in plant membrane. For the methylgesmonate, in the methylgesmonate that we apply in the post-harvest application, we found that using the methylgesmonate, they can make the plant tolerant to the heavy metal, shielding injury, drought stress, and salt stress. For the both of these stress, they found that methylgesmonate, they can enhance antioxidant systems and also activate different mechanisms in the plants. So, and then we will go to the work that I have done so far about the use of salicylic acid and methylgesmonase in the post-harvest application for the quality of the fruits and vegetables. From my study, the first one, I have done to use the salicylic acid immersions to maintain the qualities of the guava fruits. It's cultivated kimchi. It's quite famous in Thailand actually. And in this technique, we use the immersions for 10 minutes. And for the result, we found that the use of the salicylic acid can reduce the post-harvest diseases during storage, and they also maintain the visual appearance. And the 2 millimolar salicylic acid can maintain the visual appearance of the guava fruit for 18 days during storage at 13 degrees Celsius. So in this picture is also show the visual appearance of the wax apple fruit, or roast apple fruit, or java apple fruit, all the same name. In this work, we use a post-harvest immersion as well, but actually we immerse the wax apple fruit about 30 minutes, and we found that the wax apple fruit that treated with the 0.5 millimolar salicylic acid could maintain the visual appearance than the control and the higher concentrations. But actually, this shows that when you use the salicylic acid, you have to use at the proper concentration. It depends on the fruits and vegetables, because some fruit and vegetable is quite sensitive to the salicylic acid. If you use a higher concentration, you will show the toxics, and you need to use a small concentration, and it will give the good benefit to maintain the qualities. And this picture is show the visual appearance of the methylgesmonase immersions in the guava fruit, CV kimchi as well. In this work, we found that the methylgesmonase, 0.1 millimolar methylgesmonase can improve the qualities of the guava fruit compared to the control, and they also delay the rots that happen on the fruit during storage as well. So, and these slides show the effects of the salicylic acid on the textures of the fruit, and we found the salicylic acid can maintain the firmness of the guava fruit throughout the storage, and is related to the inhibit the increase of the soluble pectin and the decrease of the insoluble pectin. And do you know the changes in the firmness of the fruits mostly is related to the cell wall component degradation, especially the pectin substances. The increase in the soluble pectin is uh, enhance the softening in the fruit and vegetables. So if you want to maintain the quality, the texture of the fruit and vegetable to make it firmer, and you have to use some technique to prevent the increase of the soluble pectin and try to maintain the insoluble pectin. So in this work that I have done in a Thai papaya, it's called Kekdam papaya. And we also found the same results that salicylic immersions. We use salicylic immersion for 1 hour. And we found that salicylic immersion, about 2 millimolar salicylic acid, can delay the softening of the papaya during ripening. And it's also related with the pectin substances. And you you can see the pectin substances, the EDTA soluble pectin in the salicylic acid treated fruit is lower than the control. And when you see the sodium carbonate soluble pectin is the insoluble pectin is higher than the control. And moreover, we also found that the salicylic acid treatment is also prevent the membrane dysfunctions. We have been measuring the electrolyte linkage in the papaya tissue, and we found that the electrolyte linkage of the papaya fruit treated with salicylic acid has electrolyte linkage is lower than the control and is significantly lower. So the electrolyte linkage and or membrane dysfunction, is its effect to the texture? Yes, it is, because this membrane is related to the torque pressure of the tissue. If the torque pressure is changed, it means the softening is changed as well. So the one factor affecting to the softening, besides the cell wall modification, is the dysfunctions of the membrane. And salicylic can maintain the membrane functions. So in this picture, it shows the effect of methylgesmonase on guava fruit textures as well. And we found the similar results that the firmness is also maintained by using the methylgesmonase. And the methylgesmonase treatment also prevent the increase of the soluble pectin and the decrease of the insoluble pectin in the guava fruit. So, and then we will move to the antioxidant system and bioactive compound. The both salicylic acid and methylgesmonase, as I have talked in the previous slide, that they induce the antioxidant system and defense mechanism. So in this work, it's clearly shown that salicylic acid is induce antioxidants activities, both and FRAP assay and DPPH free radical scavenging activities. And in this work, we have done in the wax apple fruit. We dip the wax apple fruit in the salicylic acid, and we found the salicylic acid can induce the antioxidant activities, and they also enhance the bioactive compounds such as the totophenols and flavonoids as well. And moreover, we found it's not only antioxidants and bioactive compound that's induced by the salicylic acid. The salicylic acid also increase the antioxidant enzyme activity, such as superoxide dismutase, peroxidase, and catalase as well. When you see the results, in these results, you found that the fruit treated with the salicylic acid has a higher antioxidant enzyme compared to the controls. Actually, this increase in the antioxidant enzyme is related to the oxidative stress resistance in the plants during storage. So this picture shows the antioxidants and total phenolic in the guava fruit using the salicylic and methylchesmonase. It's really clear that the both treatment can induce the antioxidant and the total phenols in the guava fruit. So in this picture, we have done in the queen pineapples as well. And this work, I'm worked with my colleagues at the Chumphon province campus. And in this work, we use a salicylic acid and methylchesmonase. So in this work, we operate the results at the King Mongkut University, Thonburi. Actually, this work is like the co-work with my university, King Mongkut University, Thonburi, and the Chumphon province campus of the KMITL. So in this work, actually, we propose, we use the plant regulator to induce the shilling injury resistance in the queen pineapple. When we also study its effect to the antioxidant system. And we found that the both salicylic acid and methylchesmonate can enhance antioxidant system, and certain bioactive compounds such as ascorbic acid, catalase enzyme, and ascorbic acid peroxidase enzyme in the pineapple during storage. So, and we also studied the effects of the salicylic acid and methylchesmonate on the shilling injury inhibitions. And we also found that the salicylic acid can inhibit the severities of the shilling injuries in the post-harvest crop during storage. In this work, we study using the pre-harvest treatment salicylic acid with the hairy bacills. And we know the hairy bacills is very sensitive to the shilling temperature. If you keep the hairy bacills in the refrigerator, its shelf life is less than 4 days. Actually, 2 days, you will see the brown spot and brown flake on the leaf already. Is that symptom is present the shilling injury that happened on the hairy bacills. Compared to another basil, like a Thai sweet bacills or holy basil, you can keep it longer. So in this work, I'm interested in the hairy basil because it's very sensitive to the shilling injuries. And for the pre-harvest treatment, we found that 5 millimolar salicylic acid pre-harvest treatment, we spray the salicylic acid before harvesting 24 hours. And we found that they reduce the shilling injury of the hairy bacills, and also they reduce the increase of the electrolyte linkage and the malodaldehyde contents. The malodaldehyde content is a secondary metabolite that result from the lipid peroxidation. From this work, it showed that the salicylic acid can reduce the severities of the chilling injury by its maintain the membrane's functions. So in this picture, we show the effects of the salicylic and methylchesmonase immersions on the shilling injuries of the queen pineapples. We found that the both salicylic acid and methylchesmonase can alleviate the shilling injury symptom during storage. But anyway, from our research, we found that methylchesmonase is more effective to increase the shielding injury resistance rather than the salicylic acid immersions. And in this one as well, in this one, the effect of the salicylic acid, but we use a different technique. But actually, we know the chilling injury that happened in the pineapple, we found in the tissue adjacent to the core. And so we use another technique by using the pedicel infiltration. That means the fruit can suck the chemical directly to the vascular system into the, to the core. And we're thinking the pedicel infiltration might have more effect than using the immersions. And what do we found? We found that the use of salicylic acid pedicel infiltration show the lower shielding injuries compared to the control. And we use the positive control is the abscisic acid. But the abscisic acid has been confirmed is the plant regulator that prevent the chilling injury in the pineapple fruit. But the use of abscisic acid is not safe to the human. So thus we just use this like a positive control to compare with the salicylic acid application. And we also found the salicylic acid application is show the lower lipoxygenase lipase activity and maltodextrin content activity compared to the control. And another chemicals that we use, like a chemical elicitor, is the cyanocobalamin. Cyanocobalamin is the derivative of the vitamin B12. And we know the vitamin B12 is played an essential role in the transfer methyl group in the last step of the methionine biosynthesis and photosynthesis capacity. And we also that the B group vitamin also stimulate the phenolic compounds, proline biosynthesis that affect to the membrane. So from this, we're interested to use the cyanocobalamin to maintain the quality and improve quality of the post-harvest crop. It's my work that I published in this year in the post-harvest biology and technology journals. We use the methyl cyanocobalamin to improve the qualities of the sprouts. We use the three kinds of the sprouts: sunflower sprouts, daikon sprouts, and red amaranth microgreen. But actually, we found that the methyl, the cyanocobalamin, they can enhance antioxidant oxidant and also bioactive compound in the sprouts. That means it can improve the nutritional quality of the sprouts during storage. And we found the interesting thing in the red amaranth sprout that we found that the cyanocobalamin treatment can improve the colors of the red amaranth sprouts that by they induce the beta cyanine content in the sprouts during storage. So as we talk that cyanocobalamin might have the effect to maintain the function of the membrane, and we have started to use the cyanocobalamin to alleviate shielding injury in the pineapples. In this work, we're using the pedicel infiltration, and we also found that the cyanocobalamin or vitamin B12 can alleviate the shielding injury of the queen pineapple during storage. But we found that the effect is not good enough compared to the plant regulators such as salicylic acid or methylchesmonate. But I think for the application of the cyanocobalamin treatment, we need another technique to combine with this chemical, this might have the more effect, in the increase the chilling injury resistance in plants. But actually, in my recent work that it will be published in the fruits journals, we use calciums combined with the vitamin B12, and it showed the good effect alleviating chilling injury of the queen pineapples. That the paper it will be launched in the few months. So, and then I will move to the work that I have done in the physical elicitors. The physical elicitor that I'm studying so far with my friend and my colleagues, I'm used UV. Actually, the UV that I'm interested in, we interested in the UVC because UVC they have the high power compared to the UVB and UVA, and there is many previous work they reported the UVC application can maintain the quality as well as enhance the certain post-harvest quality of the fruits after storage, including they induce the pathogenic resistance and antioxidant system in the plant. So it's my work that I have done with my friend at Kasetsart University, and we studied in the Gelkamin mango. The Gelkamin mango, actually, we consume this mango as a green mango, not ripe mango. So the prevent the ripening of the mango is more important to the qualities of the mango in the market, because the price of the mango will decrease when the mango is ripened. So in this work, we use the UVC radiation at the 6.6 kilojoule, and we found that the UVC treatment, they delay the ripening index of the mango fruit. And they also maintain the firmness of the mango fruit during storage for 16 days. So this is another work that I'm using UVC radiations with the yellow bell pepper, and we found that the UVC treatment can alleviate the weight loss and also maintain the firmness of the bell pepper during storage for 15 days as well. So in this work is also related to the effect of the UVC treatment in the texture. And this work that we show that UVC can delay the liquefaction in the water bamboo shoots. This work that I have done with Dr. Wenbo, my colleague at the Anhui Agricultural University, China. And this work is already published in the Post-Harvest Biology and Technology journals. And we found that UVC treatment can prevent the lignification in the water bamboo shoot during cold storage, and they also reduce the increase of the texture of the water bamboo shoots throughout the storage for 8 days. And moreover, that we found the UVC treatment also prevent the greenness. The greenness is the one problem affecting to the quality and marketing acceptability of the water bamboo shoots. At the show in the picture, you can see that UVC treatment can prevent the greenness of the water bamboo shoots. So, and then we also investigate the total chlorophyll in the sample, and we found the chlorophyll biosynthesis is decreased as the concentrations of the chlorophyll in the UVC treated water bamboo shoot is significantly lower than the control. And moreover, we found that the UVC treatment also enhanced the total and also maintain the antioxidant activities but it did not affect to the ascorbic acid. That means UVC treatment with the water bamboo shoots, it can maintain as well, maybe enhance the antioxidant system, but might be related to the increase of the phenolic compounds. If we look at the ascorbic acid, it seems to have no effect with the ascorbic acid. In the same results also show in the yellow pepper and in the mango, but actually in the mango, we found that UVC can induce, can maintain, sorry, not induce, can maintain ascorbic acid contents. So, and then I will move to another physical elicitor that I have done recently. It's a ultrasound. And why I'm interested to using the ultrasound to maintain the post-harvest quality. But we know the sonication using ultrasound, they can enhance the physiological stresses in the plant, including the effect of the acoustic cavitation and microstream. And from the microstream cavity, the microstream and acoustic cultivation, it can increase the reactive oxygen species, and the reactive oxygen species that can enhance the antioxidant system and different mechanism in this plant. And I will move to another slide. This show that ultrasounds that enhance total phenolic content by they enhance the PAL enzyme, phenylalanine ammonia ligase. This enzyme is related to the biosynthesis of the phenolic compound, and they also enhance the antioxidant enzyme activity as well, again to the oxidative damage in the plants. So this picture is show the effect of the ultrasound treatment with the guava fruit. This work that I have then published in this year, and we show the use of ultrasound 40 kilohertz for 10 minutes. It can maintain the quality of the kimchi guava fruit during storage at room temperature for 6 days without any diseases during storage. And they also maintain the hardness and reduce the increase of the soluble pectin and maintain the insoluble pectin in the guava fruit. For the antioxidant system, we also found that ultrasound treatment also affect to the antioxidant system by they induce the antioxidant system, and they also enhance certain bioactive compounds such as flavonoids and terpenols. And this work is also published in this year in the Postgraduate Biology and Technology journals. We use the ultrasounds to reduce the browning in the lotus root slices. And we found that ultrasound can inhibit and browning in the lotus root slices. And if you show the graph result, it can maintain the whiteness and decrease the different color values, and the browning index of the US treatment is lower than control. And it also maintains the hardness of the lotus root slices and also maintain the antioxidant as well. It's so that ultrasound treatment can reduce the browning, and they also maintain the quality of the lotus root during storage. So for the conclusion, the benefits of the using elicitor that I have done so far, actually more than 10 years, and we found that it can maintain the post-harvest qualities and maintain the visual appearance during storage. Of course, they delay the fruit softening during storage, and they also enhance the antioxidant system and certain bioactive compounds that benefit to the human health. And moreover, you can use the elixir to prolong the shelf life of the tropical crops by its alleviate the physiological disorder caused by the chilling stress that we call chilling injuries. So finally, I would like to thank our team for this all research. In this picture, it showed my team from the King Mongkut University, Thonburi, Chumphon Province campus, and my friend, Dr. Wanbo from China, and my students. Thank you very much for your attention. Hello.
Okay, thank you very much to Dr. Suryan Supavanich for sharing your very interesting research findings and your experience in post-harvest technology, which I can see the many benefits of elicitous applications in fruits and vegetables. So now we would like to open the Q&A sessions to the audience. So feel free to submit your questions in the comment section in our Facebook Live, and I will read your question to our speakers to respond. Okay, we have one question here from Mr. Amos Yifang. Okay, one question to Dr. Suryan, is there any effects in terms of nutrition composition post-application of the said elicitors? And okay, that's the question. Is there any effects in terms of nutrition compositions in
Uh,
post-application of the means the post-application of the mentioned elicitors? Please, Dr. Suryan.
okay. After we treat the elicitor, but actually the elicitor, they will enhance in the some mechanism that they produce the secondary metabolites. And actually that we found that actually it can induce the phenylpropanoid pathway. In this pathway is related to the many bioactive compounds that happen after treatment. And actually what we found after you treat it, the effect is not effect in 24 hours, but it effect is happen later when the plant they get the strength and they really like that they got the strength and they induce the signal to stimulate the bioactive compounds, and the don't active compound we found that they have the antioxidant activities.
Okay. So that's mean the elicitors give the positive effects in the long terms on
Yes,
the fruits and vegetable.
yes, that's right.
Okay. I have just one question. I wonder how long that the effects would stay in the fruits or
uh,
vegetables.
Actually it depends on the variety of the fruit. In the some fruits it can I think the pattern of the increase is like a normal stress, but it's increased higher. And uh, like I have done in the some plant during storage for 2 weeks, it still keep maintain the antioxidant system compared to the control keep the decrease. But after that, I don't know, because the shelf life of the fruit and vegetable is already gone. People don't accept it, and we just throw it away. But no, normally, how long is depend on the variety of the fruit and vegetables.
Okay. So do we have any more questions from the Facebook audience? Okay, maybe we are going to have more questions later on. So thank you, Dr. Seren, for your sharing for today. So
Okay,
now
thank you very much.
we will continue to our second session of this webinar. Our second speaker is Dr. Chanpon Chawsap. Before we begin, let me give a brief introduction to our speaker. Dr. Chawsap received her bachelor and master degree in Kasetsart University in Thailand and her PhD in animal production from Nottingham University, United Kingdom in 2010. Today, Dr. Chawsap will share with us her experience and some more research findings on effects of selenium sources on carcass composition, meat characteristics, fatty acid composition, glutathione peroxidase activities, and also ribonucleotide content in broiler chicken. Uh, Without further ado, please welcome Dr. Chawsap.
Good morning, everyone. Sorry for a little bit late. Um, okay. The topic of my talk today, I just show one. I just want to present only one research that I have done about the selenium salt effect on carcass composition and meat characteristics of broiler chicken. These are the name of the people that contribute to this work. This work we join with Dr. Ras and Dr. Elvis from UTM and University of Putra, Malaysia. Uh, in For commercial production about chicken, there have two types of chicken. The first one is broiler. Broiler normally we raise it for meat, and the other type is layer. We raise it for egg. The research that I have done is about the broiler. For broiler production to be successful for production, it have a three important factor. The first is genetic, the second is management, and the third is animal feed. Okay, somebody help me to set up the screen. The first factor is genetic. This slide you can see the very obvious of the improvement of chicken breed from 1957. You can see the same day. Each day old chicken is just 43 grams when they hatching from egg. And 1978 is 42 grams, and in 2005 is 44 grams. Actually, it's when they start. Uh, it's not different a lot, but at 28 days, the big difference in body weight of the chicken. This one is more than at 1957 is one kilogram body weight at the same day, at the same age. You see a big improvement about the fast growth of chicken. And for at 56 days, the body weight of broiler is nearly more than 4 kilogram body weight. Date back to 40 years ago is just it's not like less than one kilogram. It's big improvement in terms of genetic. The second factor to be successful for broiler production, commercial broiler production, is diet. Corn soybean based diet is the uh, normal feed formulation for broiler. Corn is for energy and soybean is for protein source. Normally in commercial production, they use three-phase feeding for broiler production. Uh, the first phase is starter. They use approximately 22 protein percentage and metabolizable energy approximately 3,150 kilocalories per kilogram diet. And the second phase is grower phase. Is they use less than protein percentage in diet. And the third phase is finisher. Today we finished we we send the broiler to slaughterhouse at approximately 42 days age, and they use less protein concentrate in diet than the two phase. Uh, this is the normal feed form for broiler production now is they use a pellet in the pellet form. But for the starter, they need to crumble it to make it smaller before give to the broiler. Uh, to be able to measure the performance in terms of animal production, we measure feed conversion ratio. For feed conversion ratio or FCR is come from feed consumption divided by body weight gain, and we will get FCR. If the animal give lower FCR, it means the high feed efficiency is better for animal production. Another parameter that they measure is average daily gain from they can get ADG from finisher subtract by starter weight and divided by animal age, and they will get ADG. If animals have a higher ADG, it's better. And now for modern breed, like you can see from this slide, the the modern breed for broiler is very high ADG and low FCR. This slide shows the nutrient efficiency in different breeds of farm animals. Uh, you can see here the lowest one is broiler chicken. They use approximately 1.73 kilogram diet for to gain 1 kilogram body weight. The the highest feed uh, FCR for animal production is ruminant animals such as sheep or beef. They use more feed to be able to gain their body weight for 1 kilogram. Like sheep, they use 9.7 kilogram feed to get 1 kilogram body weight. This is the real data and the forecast data from 1985 until 2025 that they try to improve feed efficiency for broiler. At 1985, the broiler used 2.5 kilogram diet to gain 1 kilogram body weight, but at the uh, at the present is 2020, right? They use 1.13 kilogram diet to gain 1 kilogram body weight, and they uh, aim to the best FVR for broiler production. They just aim for 1 kilogram diet for 1 kilogram body weight. I'm not sure it can happen or not, but now this is forecast, but in the in the real situation now for broiler production, especially in Thailand, we cannot get this in commercial production. We get about 1.5 or 1.6 FCR for the use nearly approximately 1.6 kilogram diet to gain 1 kilogram body weight. Uh, three factors that I told you. The first factor is genetic, the second factor is diet, and the third factor is management. If animal have a best genetic, it have a best feed, but the best diet. If the management is not good enough, they cannot reach their their genetic potential. Therefore, in the present time for commercial broiler production, they have a very good management. They use like a closed system, evaporative cooling system to raise chicken. They keep the chicken in the closed house system and control temperature. In Thailand, we use temperature approximately 28 degrees Celsius. lower than the environment temperature. We give enough feed and water for animal, for broiler. The drawback of the fast growth animal is have some disadvantage of fast growth animal for in the modern breed. I want to say it have a two big problem for fast growth animal at the present day. The first is health of chicken because they they are very stressed because we raise very intensive in the house chicken house, and they have a stress, so they have a problem with disease. Like you can see this picture, the big problem at the moment for broiler production is they have a they're too heavy, and their bone is not strong enough, so they cannot stand up or walk properly. Is one problem that uh, nutritionists try to solve out this problem. And for another issue is about meat quality. The the big problem for meat quality about the pale soft extruded like chicken. If the paler, softer, and more extruded, they will lose more water from meat, and if they lost more water, they will lose more money because it's, they sell by weight, right? And the color is pale. It's not we cannot convince consumer to buy. This is a big problem for meat quality for chicken. And also they have problem about white strapping and wooden breast. And I have a current project about wooden bread. We try to solve solve out this problem by add some feed additive, but this project is ongoing project. I still not have a result yet. I tell you more about the PSE. This is the normal meat for broiler. It's more it's more pink, and it looks more firm, and it's not ex, not no water come off from it a lot. But this is PSE light. It's paler, it's softer, and they lost more water from meat because the protein is denatured, and they cannot hold the water in the meat, so they lost water. This one is white striping. It's a big issue for commercial broiler production as well. If you can see, this is normal breast. This is breast meat of chicken. You cannot see the white stripe, the white line on the breast muscle. But for the moderate, this score one, you can see some white line. And if score three, the extreme one, you can see the big white line on the breast muscle. Normally for meat quality, it doesn't have problem for have a white striping, but it maybe when the consumer look at it, maybe they doesn't like it, or like that. And the other problem in broiler meat is we call it wooden breast. If you can see from a what is wooden breast mean? Wooden breast it means that it have a hardness on the breast meat of broiler. This picture they use a 200-gram weight and put on the breast muscle. You see, it's not compressed, but for the normal one, 200-gram weight can compress the muscle. This how hard it is. It's very hard. If you touch it, you can feel it. It's very hard. Uh, okay. this is the aim of broiler production today. At the present, they finish the dressing, the end of the dressing broiler for 42 days, but they aim to finish broiler at 35 days. They plan to send broiler to slaughterhouse at 35 days with approximately 2.5 kilogram body weight. And to be able to do that, they need to have a nutrient balanced diet. They need to provide nutrient balanced diet for broiler. For nutrient balanced diet, what's the main composition of nutrient balanced diet? The two main composition is energy and protein source, but it still have another composition such as mineral, vitamin, and amino acids. The synthetic amino acid that's specific to use in nutrient balanced diet. Uh, the one that I did for research is just the mineral. The mineral that I interested in is the selenium. What the importance of selenium? There are many references that mention selenium have a can remodel fatty acid composition in meat. It can improve meat flavor because it can increase ribonucleotide content in meat. The main ribonucleotide that I found in the reference that selenium can improve is IMP. IMP is inosine monophosphate. The, this chemical can affect can give umami taste for broiler meat. And actually, the first start to use selenium, they doesn't interested in meat quality. They're interested in selenium can improve health of chicken and by improve immunocompetence of the chicken. And if they are healthy and if they can have a good growth performance, better performance, and give higher productivity. I will show you a little bit about the ribonucleotide content that we measure in our research. What is the ribonucleotide content? Ribonucleotide content that give a umami taste is inosine monophosphate and guanosine monophosphate. Where is this ribonucleotide come from? It comes from the ATP and it degrades during the postmortem during we store meat. Uh, it degrades to ADP, AMP, and IMP. If If it's higher IMP, it's better taste. But if degrade, if we keep meat longer, it will degrade from IMP to inosine, inosine, and finally it go to hypoxanthine. This inosine is tasteless. It is not bad actually. It's just tasteless. But if go to hypoxanthine, it's a bitter taste. It's not good for flavor. Um, for selenium, why they can improve animal health, why can improve meat quality, or they improve all these things by via antioxidant system because selenium can increase the glutathione peroxidase, the name of the enzyme that uh, very important for antioxidant system. The selenium form that they use in commercial Broiler Production. The traditional form is inorganic form. They use in the form of inorganic form that they use is sodium selenide, and recently they try to use the organic selenium because they believe it low toxicity and more availability than inorganic form. The form that the organic form of selenium is that in the commercial is the first form they use is yeast for selenium enriched yeast, and now they try to change to use like a zinc selenomethionine. They said this one is more stable and more bioavailability than selenium enriched yeast. But for our research, we didn't compare between two forms. We just compare between sodium selenide and zinc selenomethionine. The objective of our research is to compare sodium selenide form and zinc selenomethionine, and we also interested in the mix, the composition, the combination between organic and inorganic form because uh, inorganic form is cheaper, but it's uh, a little bit high toxicity and it's lower bioavailability than organic form. So we think if they can, if we can use the combination, and maybe we, the price will be lower, and we want to see the effect. If it's better, we can go for this. The parameter that we measure in the experiment, we measure the carcass composition, meat characteristic, and we measure fatty acid composition, ribonucleotide content, and also the glutathione peroxidase. We use a 540 upper acre broiler chicken, and it have a 6 replication for each treatment. In each replication, we use 60 broiler. For diet, we use 3-phase feeding diet. For starter fat, we use 21.5% protein, and grower 19 for grower and 17 for finisher. For carcass composition, we cut out the chicken, the whole chicken, and we also measure the body composition of the chicken. For meat characteristic, we cannot take all chicken in each replicate. We just take 3 birds from 30 birds. We randomly select it from each replicate. Total is 54 birds for measure meat characteristics. We measure pH, deep broth, and color and shear force. And for chemical composition and ribonucleotide contents, also glutathione peroxidase. Due to the limit funding, so we just can use 1 bird per replicate to measure these. We use GC gas chromatography for fatty acid composition, and we use kit for determine glutathione peroxidase. We use SPLC for ribonucleotide content measurement. The result from our research. For body and carcass composition, we cannot see any difference between treatments. We see a little bit higher body weight, light weight for organic. The combination between inorganic and organic form, and also inorganic form, high a little bit higher than the inorganic selenium salt. But for carcass composition, is we cannot see any differences. For meat quality, we see a big improvement for when we use organic sauce. We see the lower shrinkage loss. They lost they lost less water in organic form. But for cooking loss, it's different. The reason why the organic form is less cooking loss, maybe they have more water before cooking, so they lost more water. We just think that way. But for color, we cannot see any difference for color, pH, and also shear force value. Shear force value, if they give higher shear force, is more tougher, or lower shear force is more tender. But this form of selenium doesn't affect shear force value. For we measure cholesterol content and also fatty acid composition, we see a little bit change in the ratio for PUFA, polyunsaturated fatty acid and saturated fatty acid. If the ratio is higher, it's better in terms of meat quality. But uh, the, if if you can see from the superscript of inorganic form and organic form is better than the combination between two forms. In this study, we see a big a, a big effect of form of selenium. The organic form give a better glutathione peroxidase activity, better than inorganic form. The is a high significant difference. But for ribonucleotide content, we cannot see any effect of selenium form. For the conclusion, we can conclude that uh, selenium salt organic form can improve water holding capacity by decrease by have less shrinkage loss, and organic form can improve uh, can increase glutathione peroxidase activity. It have a glutathione peroxidase activity higher than inorganic form. So this is the conclusion of our research. The uh, sorry, maybe you have a little bit confused. The cooking loss is increased for is decreased is lower in inorganic form, but for shrinkage loss is better in organic form, and glutathione peroxidase is better in inorganic form. Uh, in my opinion, for the commercial broiler production, it's still better to use selenium in the form of organic, like metering activity. But the combination that we plan to see the effect is better effect than uh, inorganic form. Is still it's not a big big improvement. So in my opinion, I still recommend to use in the form of organic form. Uh, we would like to thank you, the Thai Food Group Public Company for research funding and my department for research facility. And these all my students that contribute to this work. Uh, some of them are postgrad and some of them are still undergrad. Thank you very much for your attention. If you have questions, please feel free to ask me. Thank you.
Okay, thank you very much to Dr. Chan Ponchosa for your nice sharing about R&D and broilers and organic selenium in chicken feedstocks. Now we would like to open a Q&A session for the audience. So, dear audience, feel free to submit your questions in the comment section in our Facebook Live, as I will read your question for our speaker here. Do we have any questions from the Facebook Live? It seems like we don't have any questions at the moment. Okay, sorry. So we have one question here to Dr. Chanpon. What caused the white strip and pale meat of the chicken? Is it something to do with iron or deposition of minerals that provided uh, during the treatment?
Oh, you mean the cost of white starting, right?
Yes, and the
Why?
pale meat.
Mm-hmm. Oh, the the reason why it happened is still not clear, but they just think because it's fast growth, so it have a like a myopathy happen in their meat. It's like that, I can say. You understand what I say? It's the problem
Is it
of this is
something?
comes from that because the fast growth of chicken and maybe it's not balance of nutrient, but they doesn't have a conclusion yet. It come from exactly what it come from.
Um, so I hope that answer the questions about the white strip and pale meat that been asked by the audience. So with that, we would like to thank you to Dr. Champon again for your talk for today. So now I would like to remind to all of the Facebook Live audience to fill in your attendance form provided in our Facebook Live for the attendance e-certification letter. All right. Now we will continue for our third session of this webinar, which will be delivered by Dr. Razauddin Muhammad Zulkifli. So a little bit introduction about Dr. Razauddin. He is the senior lecturers in our Department of Bioscience, Faculty of Science, University Technology, Malaysia. He received his PhD in nutritional biochemistry from the University of Nottingham, UK. And he also had industrial experience in AstraZeneca, Chanwood, Loughborough, United Kingdom in 2003, and also one of Halal certified trainers since 2017. Today, he will be sharing his talk entitled Halal and Technology in the first 21st century, the nutritious and quality food. So without further ado, please welcome Dr. Razauddin.
Right. Thank you very much, Ms. Chairperson, with the kind introductions. So let me upload the screen first. Right. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. And today is my sessions. I'm not going to talk about research, but pretty much in a review for the science and nutrition and also halal and tayyiban. And the content that I'm going to share with you today will be what's happening during the past, present, and perhaps the futures. And we're going to have a look at how we as a consumer and a person, individuals, were affected by the progress of science and nutrition. Right, put in outcomes a little bit because parts of this webinar is online. It is an online global what we call classroom in which some of the classes will be involved in there. One of the class that I taught is nutritional biochemistry. The students also joining this seminar. Right, this is for them. Okay, first of all, let's have a look at nutrients. Why we require nutrients. Earlier, we have our distinguished speaker from KMITL, Dr. Champuan and Paul, talking about how to improve food and how to increase the nutrient content. Okay, we'll see that our body itself, we required nutrients, not a food. The nutrient comes from food. We cannot utilize our food, but however, the food need to be broken down into small substance and compounds, which we call nutrients, in order for our cell to absorb and being used. Right. The reason we need the nutrients pretty much is to maximize our immune system, our immune functions, in order to prevent any illnesses risk from the pathogens that we expose on. Especially nowadays, we heard about the COVID-19 and also other, what we call it, diseases and illnesses. And it's also prevent deficiency if we consume enough nutrients. So it prevent the deficiency, and where we will get the nutrients level in the blood maintained at the optimum level. And this will help in extending our lifespan. And hopefully we can live happily with a good quality of life. Right, just a bit of the history of nutrition itself. So it started off pretty much before the Industrial Revolutions in the Victorian era. People grew their own foods and things like that. But after the Industrial Revolutions, we've noticed that a lot of urbanizations occurs in England in the 1860s. And what happened is over there, the shortage of food and shortage of nutrients. So the government started to think about how to improve these conditions, in which, due to the homeless and hungry people around there, they're trying to restore at least a least expensive way to keep people alive and maintain workforce to generate the economy still moving. Right, and that is the first dietary standards. Now that we have different types of dietary standards come from Canada, you've got Europe's, you've got USRDA, and these dietary standards are pretty much similar among countries, and it is based on nutritional problems and dietary patterns of the populations. Right, so as we have a dietary standards, in order to keep the nutrients ample enough in the populations, different technologies has been introduced from the past until present, and perhaps we will discuss later on the future. So if you see current improvements in technology, like has been shown by Dr. Suryan Suparvanich, you have a post-harvest technology in which now you can have an apples for 365 days. So it's on the shelf all the time. And with the advancement of this technology, such as the usage of salicylic acids, just now, that's I think it's during the 1900s, the old technologies, but new technologies like Dr. Suryan shows earlier, you've got the usage of UVA and UVC. UVA, UVB, and UVC, as well as ultrasounds to modify the phenolic contents of the fruits and vegetables. And also the progress, and also we have an investment in preservations of the food that we have. We have canned foods all over, plus also the increase of the quality and safe food. And the quality, for example, we have, if you heard about this one, iron fish, which is, I think, introduced by Dr. Ailey to the Cambodians. They've noticed that the Cambodians, they having a problem with shortage of iron. And what happened is, it will be nice if you can introduce an iron to that population. So you give a bar of irons to the what we call it, populations over there, the Cambodians, and during their cooking, they need to put in the bar of iron. Unfortunately, they feel strange. The Cambodian feel strange how a bar of iron will help them, and so he come up with an idea to make it like a shape of fish. And each time they cook, they put in this iron bar, the fish iron, and this help in improving the nutrition content, which is particularly the irons among the populations. And I think I'm back to the question that being asked earlier to Dr. Champon regarding the pale meat of the, what we call it, the chicken. Maybe perhaps we can have a look at the iron content, deposition science, because during rapid growing, we may have malnutrition, the undernutrition of them, the irons. The irons sometimes, most of the time you've got the deficiencies of iron among adolescents, because at that stage you have a fast growth, right? Right, and also we know that by modifying the feed of the farm animal, for example, the one that being work being done by Dr. Champon, which is try to improving selenium in the chicken. And when we eat the chicken, pretty much we also get the nutrient, which is the selenium, which is an important traces of minerals element in terms of maintaining our oxidative stress in our body. Next, so we know that the progress of what we call it, the food and nutrients is improving from time to time. However, there is one factors that also involve, especially people around Southeast Asians and also Asian, which is we put religion as one of an important things in our life. For a Muslim, we need to consume halal food. Halal food is something like it is a permissible food being allowed by us to eat and consume. And for a food, it is clear to have halal, which is permissible, and haram, unpermissible. And however, during a certain circumstances and factors, some halal food can be haram, and sometimes it can be makruh, which is a gray areas in Muslim. And haram items can also turning into halal in a certain circumstances. So the concept of halal and tayyiban, first is it is permissible. The other one is the tayyiban. Tayyiban is something good, everything positive about tayyiban. So it is the food has to be halal, and plus added it is tayyiban. It should be nutritious. It should have a good quality, and it is safe to consume, and it is being prepared in hygiene process. So in what we call it certifications and standards, we have different standards to ensure that the tayyiban of the food is maintained. So we this include HACCP, GMP, and also MEST. Right, let's have a look at present. So in the past, we know that what's haram is very clear and what halal is very clear. So over here, if you see on the left-hand corners, we have a chicken. Chicken, by default, it is halal. And for porcine or swine, for Muslim, it is haram. It's nothing to do with the porcine or the pigs is evil or what. No, they are not. They are fine. They are just the creatures. They are fine. Only we are not being allowed to consume them. That's it. However, the complexity of present food due to innovations, technologies, and also what we call it, the taste bud of a modern man changes every now and days. If we see, we have varieties of foods on the table now, so it's going to be very difficult for a Muslim to distinguish between a halal and non-halal. So for at least we need to have an advancement in technology as well as administrations in order to what we call it, to help the Muslim civilizations or Muslim people, Muslim community and Muslim society to really recognize which one is permissible and which one is not. Because also we know the porcine itself, it is haram, and also all its derivative cannot be consumed by Muslim. So, and both chicken and what we call it porcine, they are very sustainable source of food actually. Like like chicken broiler, just took about 35 days to be harvested, and 30% of its food that they eat convert into meat, which is very efficient. Also, it's pretty similar with the porcine itself. Like each and every single things that you found in the porcine can be used in our modern food preparations. However, we need to distinguish it. For a Muslim, we need to distinguish between them. Okay, so how to do that? First, let's have a look at the concept of halal and halal certifications. So the differentiations between halal and halal certifications. Okay, for a food that you found without the logos of halal, if you see, this is the logo certified halal from Malaysia. In the middle is the one that you will find from halal from Thailand, and on the right hand corner is the right hand over here. The right is the Alpapo Mui, which is from Indonesia. So if you find a food without these logos, the logos of halal certifications, it can be either haram, it can be halal, or it can be makruh. We don't know unless you're sure that it comes and it is being prepared as following the Sharia compliance. However, when we have these certifications, we can ensure, or at least ensure that the food is certified halal, and it is definitely halal, and it is also tayyiban. So if it is a halal certification, then it will be halal and it will be tayyiban and quality as well. So just a background a bit on what drives the halal economy itself. As we know that the sizable and growing Muslim populations throughout the world, currently we've got about 1.8 billion, and I think the birth rate and also the rate of conversions of Muslims is increasing, and it is projected by 2050 the population will surpass the Christians populations, which is the most highest populations presently. And also there is an emergence of new markets due to the awareness of halal food among countries such as in Indonesia, Pakistan, India, Bangladesh, and also China, which is they have high amounts of populations, estimated about 200, 300, 400, about 500 million in total. And so that's the opportunities for halal. And the halal global market itself projected to be about 9.71 trillion in 2025. This includes different types of industries, including Islamic finance, halal travel, modest fashions, media and recreations, pharmaceuticals and cosmetics. And the halal food itself, it's about 1.3 billion. So before this, I don't really know how much is in trillions, but after doing some readings, it is actually about 12 zeros you've got for a trillion, 12 zeros. And this is what we call a survey being done by Thomson Reuters in 2018 and 19, and showing that Malaysia is the best countries to capitalize in Islamic economy, although our population is not that much really. Right, so I've already covered a bit on the past and present histories of nutrition and also halal. Now we're going to have a look at the challenges and the futures of nutrients and also halal itself. So as we know, the productions of food itself definitely will require cost, and also how to really maintain the quality of food itself. We need to have regulations, and also the cultivations of the food. And the environment plays an important role as well as time. And it has to be balanced with our own, what we call it, way of life. Yeah, it includes our spirituals, our health, lifestyles, and populations. So let's have a look at it. So let's have a look. I'll borrow these slides from Mr. Simon Ashton. He is an financial advisor in ADs, which is a company, insurance company. They have a webinar, and I attended that webinar, and it shows that how much will life cost when you are retired, and will your money keep up with inflation? Because we know that the scientists and also the food industries try to provide as much as possible to make sure that the food is on the table. So if you see, in year 2010, burgers and fries cost about 11, and today it's about 17 ringgit. And in 2030, so it will be around 26 ringgit. 1 kilogram of salmon fish is about 20 ringgit, now 35 and 52 ringgit. For recreations, 12 ringgit, and go to 27 ringgit in the future. So we'll see that due to the inflation rate in Malaysia, it's about 3 to 4% every year. So we'll see that the inflation rate will increase from years to years, and we need to do some calculations in order for us to make sure that everybody have a good food and quality food as well. Right. So the current efforts, what have been done by the scientists, we know that earlier we discussed about how to improve the nutrient content of our food, and also looking at alternative way of cultivations. We heard about urban farmings that being what we call it quite popular nowadays, urban farming. We've got aquaponics, in which you try to grow your own food, and also trying to have a look at different alternative of a source of food, such as algae, chlorellas, and also improving the current cultivations that we have. So at present day, and also have a look at the futures a bit, what we're going to see on the market later on. So we'll see presently we have a plant-based meat. Why? Why is it plant-based meat? because parts of it, if you see earlier slides, I show you about the challenges about the what we call it the spiritual, and also about the concern of the environments as well. So does it more healthy, more sustainable perhaps for plant-based? And near the future, we will see a cell-based meat. Currently, we cannot find it in the shelf, but it's already being made for the cell-based meat. Perhaps also Dr. Saiful earlier mentions about cell-based meat in research, in our groups, BioInspira, and further questions will raise whether it's halal or not. We can discuss later on. Right, let's have a look at the present. Currently on the shelf first, we've got a synthetic meat nowadays, started in 2013, and it is being improved. Last time during 2013, it was, the meat itself doesn't taste like meat. People don't really like it. However, nowadays we've got a meat that you can't really differentiate between the real meat and also the synthetic meats or the artificial meat made by plant-based. So if you have a look at the one that we have over here, there is a comparison between Impossible Whopper, which is a synthetic meat, and also regular Whopper, the one that we found in Burger King. So we in Malaysia, we haven't got Impossible Whopper yet, but it is on the market in the US now. I'm not sure about in Thailand, but I think maybe pretty much similar like in Malaysia, not yet coming in, but it's already in the market in the US. So if we see the nutritional fact about Impossible Whopper and regular Whopper and normal Whopper, which is the beef base. So the calorie about 660 calories for regular Whopper and 630 calories for Impossible Whopper. So they managed to reduce the amount of calorie about 30 calories. To put it in perspective, 30 calories is pretty much about the 1/3 of a big banana. That's it, or about less than half a scoop of mayonnaise. And then, but however, the improvement you will see on the trans fat, which is 0% of trans fat, and the amount of cholesterol over there. However, if you see, maybe later on I wrap up a bit about this synthetic meat. Next, in the near future, perhaps we're going to see a cell-based meat. So cell-based meat is a technology is being, what we call it, vigorously studied and improved by labs, and they wanted to have like the store capable of producing their own meat. What we have over there is like they are culturing a muscle cells in cell culture conditions, and then provided it with amino acids and also the medium buffers, adding it is also based on a blood, which is can't be used, a serum which is come from blood, which is not halal for Muslim, and started to exercising, stretching the fiber muscles until it grows. Right. So what about the taste? Currently, the taste, I haven't tried and I never tried, but based on what we can see, is like it's a solely a muscle, so it's going to be a bit dry without the adipose tissues. If like a nice meat, it should have like marblings of fats in between the muscle, which give the good taste. Is it nutritious? Yes, it is. I think so. Cost effective? Currently, it is not, but in the near future, it will be cost effective, and I believe that it will be cost effective. And for the nutritious, an improvement can always be added to that. Okay. So to wrap up a bit for my sessions for today. No single food, not even a superfood, can offer all the nutrition, health benefits, and energy we need to nourish ourselves. And the 2015-2020 US Dietary guidelines recommended healthy eating patterns, combining healthy choices from across all food groups while paying attention to calorie limits. In other words, it's not only solely of the health. Our health is not totally based on the food, what food that we take. Definitely that is one of the factor, but also we need to know ourselves. We need to know our body, how our body works, and what sort of activities we need to cover if we eat a certain regimes of food and diet to keep us healthy. So a quality and nutritious food can be obtained by definitely R&D being used as a scientist. We as a scientist, we try to improve the qualities of the food itself. And same as important as that, we need to have regulators that are responsible in regulating the quality of the nutritious food, and also an ethical provider, the one that provide our food and also preparing our food, that they need to be ethical as well to make sure that what they claim is what they give to us. And last but not least is we as a consumer, a customer, we need to know what need to be eaten and what is quality food, what sort of food that you should eat based on your conditions, and what sort of activities that you do. And due to the high nutritious food and also good health care, so globally we are all living much longer. And based on this study, this research, it is expected that people who born in 2000 and 2007, they can live up to 103. For us, the old generations, we also perhaps will live up to 93 years old. Me, for example, 1977, perhaps will be live up to 93 years old. And we need to be prepared so that we're not going to be a burden to the societies, perhaps contributors. And as we know, as we live longer, more money are required to maintain. So pretty much another 30 years. Right, and we know that high medical costs, unlike a normal inflations in Malaysia, the inflations of health services and also health what we call it medications and things like that, it's increasing like 9 to 10. I think it's 12%. It's quite high compared to the others. Right, so balanced diet and balanced life are important in order for us to make sure that we live in so-called a well-being, right? And I finish or I ended my sessions with a quote saying that the tallest tree catches the most wind, the herbs dancing with butterflies and bees. We are not perfect, though society portrays us differently. Live moderate and still to keep life precious to be. And thank you very much for everyone. Now I think we'll open for the Q&A sessions.
Okay, thank you, Dr. Azauddin, for your very interesting talk, and it's very intriguing to all of us. So now we would like to open a Q&A sessions for the audience. All right, so now we have one question from Ms. Li Yifeng. The question is, will it be any genetic concern if we consume cell-based meat?
Right, okay. All right, thank you, Ms. Li Yifeng. Okay, your questions, I'm not sure what you're trying to ask. However, I'll try to answer it. Perhaps maybe it do answer your questions. Okay, concern about genetic consume cell-based meat. Okay, for actually I think it need to be clarified a bit what sort of genetic concern, because you know that we eat food every day, and each food, they are carrying genetic codes as well, right? If you see in plant cells, animal, in fact animal cells as well, they are all having a genetic codes. You have a DNA in there. So what happens once you get into the body, they are being broken down into smaller compounds. And but I'm not sure what's the questions actually. Will it be any genetic concern if we consume cell-based? Do you mean that the genetics? Do you mean them? I don't get it. The questions. Can you clarify, Miss Lee Yi Fong, regarding that? What sort of genetic concern? Because the genetic transfer normally for a human, it will be during what we call it. You get married and then you've got crossing of the genetics, or you get being infected by a virus. Virus injected the genetics into your cells, and then the genetic code, perhaps some of the virus will embed it in the DNA and cause the human genetics to be contaminated with virus. And if you see in human genetics, actually you have lots of virus genetics mixing inside us. And you know that it's only about 5% of the genetic codes that we carry in the chromosomes. The rest is just we don't know what's in there, but the one that we know that it's functioning is about 5%. Right. I hope that answered your question, Li Yifeng.
Yes. Okay, thank you, Dr. Razadin, for your answer for the first questions. Do we have any more questions from the audience? Okay, it seems like there's no questions at the moment. Okay, so again, we would like to. Okay, sorry, the question just popped up. Okay, Dr. Razadin, from the same person.
Okay.
So the question is, the cell-based meat, is it kind of clone meat from the real meat?
Oh yeah. All right. Thank you very much. Yeah, definitely. Definitely, it is pretty much like that. Yes, it is like you get what we call it muscle cells and muscle fiber. You grow it in the in the condition in the cell culture, and you nourish it with nutrients, and also you do an exercise. You give an exercise to it, stretching it in order for it to develop. So you will get like a strips, but it's solely totally a what we call a meat. Yes, it is a clone of a meat. You can see a clone of meat. So in fact, actually, if you see a meat itself, all are carrying this one slice of meat will carrying all the same genetics codes. Only that this one it will be more because expanding rather than just from one cattle, but perhaps one cattle will feed 1,000 people or 2,000 people instead. So you won't have lots of genetic variations among them. Cell based meat. However, we can always improve that. We can make a mixing inside. All right. Thank you.
Like modifying the characteristic or properties of the meat.
Yes,
the new meat that we are trying to produce, maybe. Okay.
Yeah.
Okay, Dr. Razadin, there's another question. I think we got ample time to get more questions here. So from Mr. Amos.
Okay.
Could cell-based meat cause disease such as cancer? So I think this question probably that reflects to the first question of the genetic concern just now.
Yeah, thank you very much, Mr. Amos, regarding could cell-based meat cause disease such as cancer? That is a $1 million question, which is we are unable to answer that. Even now, we still try to find the cause, because we know that cancer itself is multifactorial, right? In the cancer, we know that parts of it is genetics. We also know that parts of it is our immune system. If your immune system is good, then it will suppress the cancers itself. It's only during like what we call it when our body is not fit, and then it can generate cancer. Could cell-based meat cause disease such as cancer? The answer is yes and no. Yes, without the cell-based meat also, we can get cancer. Introducing it into our diet, perhaps yes, it is. Cancer is something that we cannot run. It's already in us. Whether it's going to be expressed or not, it's up to us how we live our life. And also sometimes we try as much as possible having a very good diet, a very good healthy exercise and everything. But again, a good genetics perhaps, yeah. But again, sometimes it's also happened. Like in insurance company, they always calculated that 80% is human risk. Yeah, 80% is human risk of what we call it a human risk itself, because we make mistake in things like that. 18% is a failure of equipment. There is 2% act of God. We don't know. You keep healthy, keep everything, but if you can, you're being knocked down by drunk drivers, for example, or and that's the fate that's why some of us still holding on with the religions. I hope that answer your questions regarding the disease and cancer.
Okay. So we have another questions. From Li Yifeng, there's another maybe another Q questions after this. Will it be able to produce a cleaner meat than the real one?
Right. Regarding the cleaner meat and the real one, I think we need to look at things from different perspective. Why do you consume the meat itself? Is it for pleasure? You want it to taste good? Or is it for growth? Yeah, growth, your muscles and things like that. And also, is it for your health and spiritual, based on your religions? For me, I think I will answer it for all these factors, right? So it is not definitely you will produce a cleaner meat. I believe cleaner meat is without without the fats itself, but the fat is the one that gives you the nice taste, and you can always burn the fat enjoying the food, rather than just eating just a sole proteins, which is you're not enjoying your food you're eating, right? So will it be able to produce a cleaner? Yes, definitely. It is a leaner meat. It is a leaner meat when compared to the meat that we have, because it is 100%, almost 100%. It is protein. You don't find adipose tissues in there, except for nowadays they try to improve it by adding adipose tissues to make it feel nice. People pay a lot of money to eat Wagyu. Why? Because Wagyu is very nice. You have a marbling adipose tissues in between the meat that give the nice taste. Okay, thank you very much.
Okay, that's the next question is from Mr. Rushaidi. Is cell-based meat categorized as dead meat, since dead meat are forbidden to Islam?
All right, thank you very much. Very interesting questions. So the cell-based meat categorized as dead meat, since dead meats are forbidden to Islam. Okay, if you see from the root, we have to go into the beginning, the roots and the process. So if, for example, the meat itself being slaughtered based on the Sharia compliance, and the meat itself is halal to be consumed, right? And the derivatives of it is also halal. So, but next, you have to have a look at the process involved. When you culture a cell-based meat, you will put in a serum inside that, and the serum itself, you cannot use serum. You cannot use serum, and that's why it's not going to be permissible for that. And yeah, from my point of view, however, we don't know, because if you see from the hierarchy of Maqasid Syariah earlier, I shared with you, we need to have like a Sharia discussions among them, the scientists, and also the what we call it, the religious council to really mentions to really make a decisions about that. But in general, I think it won't definitely because of the process itself. If you replace a different process, it will be a different story. Further discussions can be looked at. Okay, I hope that's answered that question.
That's very intriguing questions from Mr. Rushaidi just now. I just if I can just share my thought, because is something that meat is something that from the live animals that we have to slaughter first, and then with certain procedures that to make it halal. But in terms of cell-based meat, it's a different process that we actually culture the cells that will multiply and divide it into tissues, and be the meat. And I'm not sure if it can be said that there's no life, I mean, in the tissue, instead of compared to the real animals of the real chickens that running away, and you have to slaughter properly, compared to the cells that we grow in vitro. But again, I really agree with Dr. Razaldin that we really need the more Islamic professional to say into details to make this kind of decisions. Do we have the next questions, please? Yes. So when we are having the cell-based meat, will it be high chance for us to consume chemicals as well? Since the meat are growing in petri dish, I think that's reflects to the process that we are providing the nutrients, obviously the chemicals, to make sure the cells and the muscles that we are producing in the lab, is healthy and safe to be eaten. So I think that's the questions, whether is there maybe there's post process like cleaning up the chemicals during the production process, or I don't know.
All right,
yes,
thank you very
please.
much. Okay, let's have it. All right, in order, I try to try to understand the question first. So you are saying that, okay, for us to consume a chemical which is being used for what we call it, producing the meat. That's your questions, I think. All right. Currently, we know that for a cell culture, we do rely a lot. Not rely a lot. We rely on antibiotics. All right, like it or not. Even in the farms as well, we use antibiotics. But over here, I think the amount might be higher, but you will always have a process to you can always remove the antibiotic process in order to do that. Yes, definitely we use chemicals in order to grow it. Except for like if you grow a cell-based meat over there, it takes you very short times, like broilers 35 days. So 35 days, the same amounts of meat, a lean meat, the total meat without the feathers and also without the bones, and you get like a lot of people that you can feed with this technology. But again, it is not. I think from my points of view, it is not the whole package that you have in natural, because we have a law being implanted in each of us. If you are a person, a believed person, in which all them being in this world, they will try to find as good as possible. If you let them, the chicken, eat, they will try to eat the best food that they have, and we will eat that chicken and we'll have the best nutrients, pretty much like that. But over here, we are introducing different things. So and to cut it short, it will be like yes, it is. Yeah, you have chemicals in there as well, but you can always remove it. But adding a process definitely, and adding a cost, and so we have to balance out between the cost and also then the meat itself. Okay.
If I can add some more, but again, the
Sure.
process, you know, everything has chemicals in it. Even the natural food that we eat, it's just a matter of the concentrations and the safety of the chemicals itself.
Yeah.
I think
Yeah.
if that would give
Yeah,
a
thank you.
clearer idea.
Yeah, that's why it's very good for us. It is very important for us to look after our water, look after our land properly, our soil, so it is not contaminated with unwanted things. So if we have a high concentration in certain things, and then we have a food that absorb lots of these particular minerals or particular heavy metals or particular chemicals, and if you consume it, then it's not going to good for us, and then there is lots of question about disease and cancer and things like that. Okay. Right. That's why if you see in them earlier, I mentioned about the regulators, about the providers and things like that. So if we have everybody's playing their parts nicely, so we live happily, well-being,
Inshallah.
inshallah.
Okay.
All right.
Right. I think that's all for the quick N&A sessions for the Razaudins. And again, we would like to thank you very much to Dr. Razaudin for your interesting sharings and discussion with the students and audience. So now we would like to move on to our fourth sessions and the final sessions for today. So our fourth speaker is TS Dr. Shazwani Itri Amran. Dr. Shazwani received her honor degree in 2008 and PhD in 2016, both in pharmaceutical science under the supervision of Prof. Philip Thompson and Dr. Ian Jennings from Department of Medicinal Chemistry and Drug Actions, Monash University of Pharmaceutical Science, Parkville, Australia. So currently, Dr. Shazwani holds senior lecturer positions at Department of Biosciences, University of Technology, Malaysia, and her research interest deals with in silico and in vitro drug screening, ADMET, OX and analysis, site-directed mutagenesis, and structural biology. Today, she will be sharing some interesting topic with us entitled From Plants to Pill, Application of Computational Tools in Natural Product Medicine. So please welcome Dr. Shazwani.
Okay, Assalamualaikum. Thank you to Dr. Norashikin, moderator for today. And today I would like to grab this opportunity to thank the organizer and also having the chance to share some research and also not so research based but more on learning learning of new things. Okay. So what I'll be sharing with you today is how do we transform plants such as natural products into medicinal products using computational tools. Okay, as you know that natural products, including plants, animals, minerals, they have been the basis of treatment for human diseases long time ago. And historically, the majority of drugs have been generated from natural products, such as the secondary metabolites or any bioactive molecule extracted from the plants, predominantly plants. And in the past, people started consume plants and natural products to treat ailments, but without the knowledge of the dosage, how much should they consume, the mechanism of action, how do these medicinal or traditional natural products act in the body, whether or not these products can impose toxicity to our important organ, or even they don't have any knowledge on drug formulation. So some of the modern medicine arise from some of our modern medicine today arise from ancient medicinal practice. And even, for example, here on the slide, digoxin is one of the bioactive molecule extracted from Digitalis lanata, is a compound used to treat heart disease. Same goes to aspirin, extracted from willow bark, and morphine or codeine derived from poppy seeds. So this has been used long time ago as pharmaceutical agent. Okay, the process of drug discovery and development is very challenging, time consuming, and also very expensive. And it is divided into several phases: drug discovery, drug development, and also drug delivery and regulation process. So under this phase, there are many other small steps need to be taken in order to complete the drug development pipeline. Started off by identifying the target. So our molecules in the body, such as proteins, enzymes, receptors, hormones, cell membranes, could be the target of any drugs. And these biomolecules are also associated in the development of disease. And usually when we look for drugs, it comes from any chemical compounds derived from lab, or even we can extract bioactive molecules or secondary metabolites from plants. Okay, so for example, if you have one plant that you send for extraction, it can give you like 10, 20, 30 compounds or bioactive molecules, and we never know from these 20 or 30 compounds which one can give you the desired therapeutic effects that you are looking for. So identifying target and also screening of ligands is one of the steps under drug discovery in order to get the best hit or the best compound that give you the desired therapeutic effect. And once we get the best hit, then we can proceed with drug development. And under this stage, we can consider the formulation, the pharmacokinetics, how safe it is when it is consumed by the animals. What about the absorption property? What about the distribution? Can this compound get to the site of action in the body, and so on? So this will be included under in vitro testing and also preclinical trials. And after we completed preclinical trials, then only we can proceed with clinical trials where human subjects are used to consume the drugs and see whether those drugs can exert therapeutic effects before we can apply for approval. These drugs are very strict, need to comply with strict regulations. And some of the drugs in the market, there are cases that some of the drugs in the market need to be withdrawn from the market due to adverse effects and so on. Okay, and the whole procedure, the whole pipeline took around 15 years and at the expense of, if not billion, million of dollars. So again, the process of drug discovery and development is very expensive and it's a very long process. So let's narrow down into drug discovery stage. So under drug discovery phase, we have a few more steps. And from step 1 until in vitro or in vivo studies, it might take around 3 to 6 years. Okay, just now I mentioned that the whole pipeline might take you around 15 years to develop a drug, but still, after you complete that 15 years, after you complete all the process in the pipeline, there is no guarantee that you can get a successful drug candidate. Okay, however, this drug discovery stage itself might take around 3 to 6 years, which is equivalent to like one PhD project. Okay. So in this stage, we need to study the target. Okay. It can be protein, it can be receptors, it can be transcription factors, hormones, ion channels, and so on. We need to understand how the protein looks like, what are the binding sites available on the protein, what is the behavior of the protein, and so on. Then we also need to find out or identify what are the ligands or compounds that we want to screen. Usually this lead discovery comes from extraction of natural products. Okay. As we know, in Malaysia, we have a lot of trees. Even there are still a lot of trees that are not discovered yet. Okay, so extraction of plants can give you data, what kind of bioactive molecules available in that kind of plant, and whether those plant has any pharmaceutical benefits or not. So we can, after extraction, we can screen those compounds against our target to look for any desired pharmaceutical properties or activities. And let's say we are fortunate, we find out a few good hits, and we use the structure of that compounds, or we call it scaffold, and make some modifications so that we can improve the binding affinity, we can improve the activity, the potency, or the selectivity towards the target. And that comes the medicinal chemistry in the picture. Okay, so in this phase, rational drug design is very important. This is to make sure that we find a good pharmacophore for the drug that can give you the desired activity, improve potency, in order for you to produce a good drug candidate. Okay, and after that, after you have modified your drugs, you need to undergo a lot of biological testing, cell-based assay, tissue-based assay, especially in vitro studies, to see how much cell inhibited, how many activities you got, and so on. Then only you can proceed testing the drug in animal model. Some animal model modeled based on the disease state. So let's say you have a diabetic mice. So you have a compound claim for anti-diabetic property. If you instill that compound into this diabetic mice, will that diabetic mice lose weight or decrease in adipose tissue and so on? If it shows that way, then it shows that your compound is efficacious, is potent, and so on. Okay, and it's suitable for anti-obesity agent. So those process will take up around 3 to 6 years. That is the traditional drug discovery process. What I just tell you just now is from screening natural ligands, you can screen out natural ligands using high throughput screening method. Usually you use 96 well plates. If you have hundreds of compounds, you might use hundreds and maybe hundreds of 96 well plates, or maybe you use 372 well plates and so on. Those are high throughput screening, and it is very laborious. You need to have a lot of reagents, you need to have good machines and so on. So once you screen using high throughput screening method, you identify your hits, then you need to modify your compounds, you perform rational drug design, and those new compounds or those modified compounds undergo another round of screening and to get the most the better compounds. Then only those selected or promising compounds can proceed into in vitro, in vivo studies or preclinical studies. So there's a lot of ways, a lot of things to do in the middle in order to identify a single promising drug candidate. And according to traditional drug discovery process, this is very laborious, very expensive, because you need a lot of expensive reagents, consumables, very time consuming. You need to repeat, need to produce duplicate, triplicates data and so on. At the end, you will end up with low success rate to get hits. Okay, and this is where computer-aided drug discovery comes in the picture. Okay, we can replace most of the methods or approach in the traditional drug design using computational modeling or computational tool. Starting from screening natural ligands to identifying target. Then you want to do screening, you want to see how well the ligand bind to the target. You can do molecular docking. And then you find good hits, you want to modify the pharmacophore. You can, instead of going to a chemistry lab, you do synthesis. You can also do that in computer. You synthesize new compounds with new modifications, and then you test it again, do another round of molecular docking and so on. Then only you can come up with a list of compounds with best hits. Okay, and then only your lab work begins. So you can save a lot of time by just applying computational tool at the beginning of the drug discovery phase before you enter the experimental study. And computer-aided drug design has been proven time efficient. It can cut a lot of costs, especially on your lab work, because it only utilize computational tools. Of course, you need to have at least one computer, very powerful one, and the success rate is high. Okay. So in modern drug discovery process, the whole drug discovery process is now replaced by computational aided drug design. And from 3 to 6 years, you can cut it down to 1 to 2 years time frame. Okay, and in fact, you can proceed on to perform in vitro, in vivo studies and so on. For example, like target selection, we have a lot of databases. We have PDB. You can get all sort of structures from there. Or if not, we have BLAST NCBI database where you can get all genomic sequences. You might go to UniProt database to get all the protein sequences, and then from these sequences you can model out your protein structure and then validate it. And then once you already have the structure of the protein, you can screen like your compound library. Let's say you have a compound library from an indigenous plants in Sabah or Sarawak, and then you want to see what are the potential pharmaceutical activity of this plant, you can screen those compounds against your target, and you can use you can just choose variety of docking programs online, and then you identify which part of the ligand, which part of the structure, or we call it pharmacophore, that contribute to the activity of the ligand, and then you can modify the compound. You make your own drug library, and then you can optimize using structure activity relationship and so on. Then only you have narrowed down your hits, and that hits can be tested in vitro and in vivo studies. This is where your experimental work starts on. Okay. So in drug design, the principle of designing drug is, first, you need to have a ligand that can bind better with the target. So that refers to the affinity. How strong the ligand can bind to the target refers to affinity. And whether that binding can exert the desired activity do you want, let's say inhibition or activation, that is what we call potency or efficacy. And also how well it can bind to the target without binding to other molecules. That is what we call specificity or selectivity. Of course, you want a compound that binds specific to your target, because if it is able to bind to other proteins or other molecules, it might impose adverse effect or toxic effects. So selectivity is also another characteristic important in drug design. So this affinity and potency can be analyzed easily using in vitro. However, specificity or selectivity can be improved with computational tools, because you can look and visualize the interactions between the ligand and the protein at atomic level. Okay, now I will give you an example of a research project that we perform using computational tools. So in the development of selective PI3 kinase inhibitors. So as a background, PI3 kinase enzyme is a mammalian enzyme. It has three classes, class 1, class 2, and class 3. And under class 1, it has 4 different isoforms. It has alpha, beta, delta, and gamma. And each isoform carry out different function. For example, the alpha involved in cellular proliferation. In beta, it involved in platelet aggregation, and delta and gamma involved in immune cell regulation. So aberration in each of these isoform will lead to different types of diseases. For example, in the alpha isoform is highly mutated in cancer, because the function of this alpha isoform it involved in cellular proliferation. So mutation of this alpha isoform will lead to development of cancer. And same goes to beta. It will induce heart disease. Meanwhile, delta and gamma will involve in autoimmune diseases. So if we can have a ligand or a compound that targets specifically to the alpha, that compound can be a potential anti-cancer agent. Okay, but if that compound is able to bind to alpha isoform, but at the same time can bind to other PI3 kinase isoform, it can also exert anti-cancer activity. However, it might have effects on the heart and also on the immune system. So this might lead to adverse effects. So that is the reason why we want to try to develop a selective inhibitor for PI3 kinase. Okay, so historically, the history of PI3 kinase inhibitors, it started off with a natural product. We call it wort-mannin, and this is extracted from a fungi called Penicillium funiculosum. And after we screen wort-mannin against PI3 kinase enzyme, it turned out to be a very potent inhibitor for PI3 kinase enzyme with 5 nanomolar inhibitory activity. And from that on, there are many researchers try to produce new compounds, and some of these compounds showed selectivity towards alpha isoform. This compound showed selectivity towards beta isoform, and these two compounds are selective for gamma and delta isoform. However, there are also compounds that are not selective to any of the isoforms. And looking at the literature search of the IC50 values, we can see that, for example, this PIC75 inhibitor can preferentially bind to the alpha isoform with very low IC50 value compared to the other isoform. However, GDC0941 can bind both in alpha and delta isoform. So the reason why we need to modify the chemical structure is because if we modify the structure of GDC, can it retain the preference to alpha and lose binding to the delta, so that this compound can be a good anti-cancer agent. Okay. So one question that we can ask from here: what makes the inhibitors selective? So we use computational tools to understand the selective mechanism of these inhibitors and how to design future isoform selective inhibitors. And here comes the computer-aided drug design again. We have our natural ligands. We have our ligands in hand, and we can construct the chemical structure either by downloading from PubChem database, or we can draw the ligands from any drawing programs like ChemDraw, Avogadro, and then you can optimize the structure using Gaussian and so on. You can also download the structure of the protein from the PDB. So in the PDB, we have structures of the alpha, beta, gamma, and delta. So that makes works easy. And then we do docking to analyze the ligand target interaction. We analyze the binding profile. We analyze the affinity of the binding. We analyze the types of bonds available, the distance of the bonds, and so on. And then we modify the structures and do another round of molecular docking and see any changes on the affinity and so on. Okay, those structures or those ligand target complex that shows a good results, we can proceed with molecular dynamic simulation to see how the structures and the ligand behave, whether the binding between the ligand and the protein is reversible or irreversible. If you want a long-lasting drug action, maybe you will look for irreversible binding mechanism. If you want a short action of drug, you might looking for reversible binding type of reaction between the ligand and the target. And we managed to narrow down a few compounds to proceed with in vitro and in vivo studies. Okay, this is the structure we get from PDB. It is the gamma PI3 kinase enzyme bound with ATP. So this is the ATP molecule. So we study the structure. Somehow we know that the ligand bind at the ATP binding pocket, and we study the ATP binding pocket. We found out that there are a lot of empty spaces which are not occupied by the ATP. So we think that maybe if we can modify the structure and occupy the space here, space down here, or even go deeper into the hinge region, maybe we can produce highly selective inhibitors. And when we study different other crystal structures, for example, this one is still the gamma PI3 kinase isoform, we found out there are two regions within the ATP binding pocket containing non-conserved amino acid. So when the ATP binds, this amino acid stays in this way in region 1 and also in region 2. But when different inhibitor bind to this space, we can see that the side chain of this lysine sways away from the inhibitor. Same goes to region 2 when we have this inhibitor PIK39 bind to the active site. We can see that this methionine side chain opens up and make a new pocket which occupied by this inhibitor, and that could be the reason why PIK39 is more selective than LY294002 compound. And when we compare the gamma and the alpha crystal structure, we found out that these amino acids, they are non-conserved within the active site. And these non-conserved residues, we hypothesize that if we can make the ligand can make interaction with these non-conserved residues, that could be the key of selectivity of these inhibitors. Okay, we also do sequence alignment across the 4 different isoforms, and based on this study, we found that this valine amino acid is critical for inhibitor potency, and the non-conserved amino acid in red boxes are those that interact with the inhibitors. So they can contribute to selectivity by interacting with this non-conserved amino acid. Okay, so what we did with our library of compound is we perform molecular docking simulation, okay, and we rank this ligand according to free binding energy, and you can see in these red boxes, the blue one, compound 16, in blue, has binding free energy of -7.6, which is lowest as compared to the green structure, compound 15, with only -5.3 kilocal per mole of free binding energy. So the lower the free binding energy, meaning the tighter the binding of that ligand to the target. So the lower the binding energy is, the better. So we chose that the blue compound is our best hit. And we took this blue compound and modified the structure, okay? And we perform what we call quantitative structure activity relationship analysis. So on the very left-hand side, compound ZSTK is the reference compound. This is the control. So we try to modify this compound. We try to remove the difluoro bit over here. We add some substituent at this side, and for this compound, we just add nitrogen, and it found out that this modification didn't contribute to increase in affinity because the free binding energy is increased to -4.9 to -5.5. So it loses the binding affinity to the target. However, when we do some modification in this compound, we remove the difluoro but add in the hydroxyl group, we can see that the free binding energy value maintained, and the estimated inhibition constant also similar to the control. And modification at the right-hand side of the molecule with a bigger substituent loses the affinity to the target. So this is not a good modification. So we will proceed with the best compound as our best hit and do more modification and do more docking studies to see whether the free binding energy value can be lowered down, as well as the estimated Ki value can be lowered down or not. Okay, so again, I show you the same slide. Everything is done using computational, and and finally we narrowed down to the best compounds that we confident to proceed with in vitro and in vivo studies to see whether this compound can really inhibit PI3 kinase enzyme or not. Okay, apart from the drug discovery process, which you can replace from doing in vitro experiment to computational tools, drugs that hits or compounds that are promising to proceed to the next stage need to undergo analysis for drug likeness. Okay, we want to know whether that compound has all the drug-like properties or not. So this can also be done using computational tools. And apart from that, we also interested to see whether that compound can be absorbed by the body so that it can get to the site of action, can be distributed in the body, can be metabolized by the body and excreted out so that that compound won't accumulate in the body and causing adverse effects. And we also want to know whether that compound can induce toxic effects to the main organs like liver, heart, and so on. So all these characteristic or properties can be predicted computationally as well. So this is one of the example how we perform pharmacokinetic prediction, okay, using this program. So you just draw out your structure and it will give you all kind of data. So you just plug out this data, and for example, this lipophilicity will reflect on the absorption and excretion property of the drug. Under pharmacokinetics, you can look for GI absorption, blood-brain barrier permeability, which indicates how well the compound can be distributed. And this part over here can predict how well the body can metabolize and excrete the compound. And this part over here, how well the drug or the compound comply with drug likeness properties. And also we can use another program called Toxicity Predictor to see whether that compound can impose any other toxic effects to the liver. For this particular compound, it doesn't impose toxicity to the liver, still considered low toxicity in cancer development, and also won't induce lung infection. So this drug somehow can be concluded or claimed as safe for consumption according to the prediction. So one might ask, what are the relationships between the in silico and laboratory testing? So which one should we start first? Should we start with our lab work or should we start with our computational work? So this can be done either way. Depends on your condition. If you have good funding, then you might start with with lab works, and then you can use in silico approach to validate your experimental results. But if you are working with a tight budget, so you can start off with computational work extensively and then select out those promising compounds or or promising data to proceed with laboratory experiments. Somehow, you might just need a high spec or powerful computer for this. So let the computer do everything for you. So that is how it works. So either way is okay. So computational biology, in summary, it's an in thing now. So we are in the fourth industrial revolution, whereby most human skills are replaced by automated or computer technology. Same goes to our biology research. So many researchers have collected data from lab experiments, and most of it now are documented in databases. So we can use computational biology, which includes many aspects of bioinformatics, to in order to understand biological system and relationships. So we can in fact build computer models of biological systems, and also use this model, um, allow for prediction of how system will react under different environments. And apart from that, since we have a growing knowledge of biological structures, if you have increasing computer power, will be helpful for you to do a lot of computational analysis effectively in various phases of drug discovery and development pipeline. So the importance of in silico tools now is greater than ever before, and has advanced many aspects in pharmaceutical research. So, but why not we apply more computational tools in different aspects, such as in microbiology, um, molecular biology, and so on, because this could give a good insight, and also good data, uh, and also can contribute more knowledge in biology. So I would like to acknowledge important people in my research journey. So we have Extraction Natural Compounds Group from UTM. We also have Medicinal Chemistry Group from Monash University and also from Chemistry Department. We have Biochemistry and Molecular Modeling from three different institutions. We have in Monash, we have in UTM, UIA, and also in Saudi Arabia. And also we have crystallography and structural biology experts from Johns Hopkins University, US. And most of the computational work here is supported by Malaysian High Educational Ministry, Australian government, and also Saudi Arabia government. So I hope you enjoy the talk. Thank you for listening.
Right. Thank you very much, Dr. Shazwani, for your very fascinating sharing. Now we would like to open a Q&A session for all the audience. So feel free to submit your questions in the comment section in our Facebook Live, and I will read it to our speaker here. Do we have any questions? Okay. It seems like we don't have any question from the audience at the moment. Perhaps maybe students who are joining this webinar can meet Dr. Shazwani during the class and discuss further about the topics. So we would like to thank you again to Dr. Shazwani for your talk for today.
Okay, thank you, Dr. Narashkin.
Thank you. So now we would like to say thank you to all the speakers for your wonderful sharing today, and I hope all the audience get the useful insight and benefits from today's webinar. Now we have come to the closing of our webinar sessions. I would like to invite the Director of Bioscience Department, Faculty of Science, Associate Professor Dr. Shafina Shahir, to give her closing speech of this webinar. Please welcome Dr. Shafinas.
Sorry. Okay. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon. It's almost 12. I would like to welcome everyone to the closing ceremony of this webinar on future of agriculture for the 21st century, organized by University of Technology Malaysia in collaboration with King Mongkut's Institute of Technology Lad Krabang, Bangkok, Thailand. First and foremost, on behalf of BioInspira, we would like to express our heartfelt gratitude to the profound speakers on sharing their research knowledge and experiences, and of course to the participants for joining this webinar. May this program mark the beginning of future sustainable collaborations between parties. I would also like to take this opportunity to congratulate the program team, BioInspira RG Frontier Material Research Alliance, and staff from Faculty of Science for successfully organizing this webinar. And also, most importantly, to our counterparts in KMITL. Last but not least, to all the participants. Thank you for joining us, and we hope that you have taken with you a lot of new knowledge. And congratulations again. Stay safe, everyone, and have a wonderful and productive weekend ahead. Assalamualaikum, and thank you.
Thank you very much to Associate Professor Dr. Shafinas for your very nice closing remark and speech. And thank you for all the participants and audience who with us throughout this webinar on future agricultural technology for the 21st century. And we hope to see you again next time. Thank you.